Influenza A virus surface proteins are organized to help penetrate host mucus. Influenza A virus enters host cells and evades mucosal immobilization through a combination of attachment and detachment proteins, which facilitate sialic acid binding and cleaving, called hemagglutinin and neuraminidase. Sialic acid is a cell surface component present in excess on a variety of cells and in the mucosal regions. Genetic changes in HA and NA glycoproteins are responsible for seasonal influenza, and novel combinations between the two can potentially result in pandemics. Although there has been a substantial amount of research conducted with consideration for the genetic basis of these proteins, the goal of this study is to characterize how HA and NA influence the mobility of filamentous virus on cell surfaces containing sialic acid. This was accomplished by utilizing site-specific fluorescence. Microscopic observation revealed a disproportionate and polarized distribution of HA and NA on the filamentous surfaces, resulting in binding and cleaving interactions, which propelled the virus away from its Na ridge poles. The pattern of binding and cleaving established by HA and Na resulted in uniform directional motility. This process serves a paradoxical role by enhancing the virus's ability to evade mucosal entrapment, while also simultaneously allowing viral attachment to potential host cells, which may provide an explanation for the prevalence of filamentous phenotype in clinical isolates of IVA. In the study, genetically modified strains of IAV incorporating site-specific tags were developed to better understand this occurrence. Engineering site-specific tags in an amenable IVA protein were accomplished by identifying sequence-specific sites vulnerable to variable labeling molecules for each viral protein. The virus propagated for the purpose of this study were derived from AWSN 1922 cells, which were genetically altered to replace the WSN M1 gene with a gene which establishes filamentous phenotypes. Cultures containing HEC293T and MDCK2 cells were transfected with plasmids encoding IAV genome segments and propagated in MDCK2 cultured in the conditions outlined in slides 4 and 5. Qualitative visualization of HA and NA-directed virus motility was accomplished through photobleaching, which concentrates an evanescent wave of energy at a sufficient length filamentous virus appropriately positioned within the field of view. The viral particle was bleached at maximum laser power and imaged in Figure 1b and Figure 2b pictured in this slide. Preparation of samples for storm imaging was accomplished by adhering antibody and sialic acid bound virus to cover slips for 1 hour at 4 degrees Celsius. The resulting cover slips were incubated with dragon green streptavidin-coated beads. The viral bound beads were affixed to microscope slides, washed 3 times with supplemental buffer. Stochastic optical reconstruction microscopy constructs a composite image from frames capturing stochastic activation of individual fluorophores with photoexcitable properties. The photoexcitable components and conditions for imaging of HA and NA are listed within this slide. In the NAI challenge assay, viral particles were propagated in polarized MDCK cells existing in a monolayer on a collagen gel. Centrifuge virus was then resuspended in viral growth media, supplemented with or without oseltamivir carboxylate and treated with 1 unit per milliliter of NAI post-incubation at 37 degrees Celsius. The challenge assay was completed in order to determine whether spatial orientation of HA and NA glycoproteins provide functional significance to antiviral challenge populations. Spatial organization was defined as the HA-NA separation between center of masses divided by virus length. Viruses that were able to evade the cell surface exhibited increased HA and NA polarization. Sialic acid conjugated cover slips and viral particles for viral motility assay were prepared under the conditions listed in slides 8 and 9. The prepared cover slips were inoculated with virus and visualized via TIRF microscopy. Motility was assayed by visualizing morphological characteristics, including channels, viral position and intensity of lectin-stained sialic acid cleave trails. NA activity was quantified through fluorescent microscopy, where cleavage of MuNANA by neuraminidase releases the fluorescent molecule methyllumbyliferone to determine differences between wild-type NA and NA delta CT. The results yielded an estimated 6-fold difference in total NA content between viruses with wild-type NA and viruses with NA delta CT. Viral imaging at air-liquid interface of Cal3 cell cultured were accomplished under conditions outlined within this slide. The production of mucosal layers 1 to 10 micrometers thick provided an interface for viral binding. Alignment between HA and NA axis were defined as displacement between center of masses for HA-NA. Displacement was defined as the vector displacement between the center of masses for HA and ECL tracks. In the results section, graph 1A illustrates polarized NA enrichment in microscopically observed fluorescently labeled HA and NA proteins as measured by relative intensities of HA and NA within 400 nanometers of each viral pole. Graph 1B illustrates polarized NA enrichment in microscopically observed fluorescently labeled HA, NA, and MP proteins, where across 20,344 particles, approximately 1/3 have NA 5-fold higher intensities at poles proximus to MP foci. Graph C and D illustrate storm reconstructions of filamentous virus at 30 nanometers. Graph E plots spatial correlation coefficients from 24 regions of filamentous particles. Graph F illustrates how HA-NA distribution affects viral cell surface escape after the addition of exogenous sialidase in the challenge assay. And graph G plots the median distribution of HA-NA polarity in filamentous viral particles in the presence and absence of exogenous sialidase. Graph A illustrates how fluorescently labeled filamentous viral particles interact with high-density sialic acid substrates. Image B illustrates a time series which shows directional tropism conferred by HA binding the fluorescent ECL track precipitated by NA cleavage. Graph C plots mean squared displacement against frequency to determine the viral diffusion coefficient. Graph D illustrates viral trajectories assembled from time-lapse data, and graph E positively correlates HA-NA polarity with viral motility. Image A illustrates differences in ECL track persistence between wild-type NA and NA delta CT. Red arrows indicate terminal viral tract dissociation. Graph B represents data from 3 biological replicates which illustrate mean squared displacements for mobile wild-type and NA delta CT. Histograms C and D illustrate experimental and simulatory directional displacement measured in angles for wild-type NA pictured in magenta versus NA delta CT pictured in green. Illustration E demonstrates the effects of sialic acid distribution on viral mobility and diffusion. Image A illustrates mucin formation as Cal-U3 cells differentiate. Image B illustrates diffusion patterns exhibited by labeled viral particles bound to mucosal surfaces. Image C illustrates ECL track formation, where polarization is apparent. Image D illustrates diffusion patterns sampled from 3 replicates that feature large filamentous particles with broadly distributed NA. Graph E correlates HA and NA axis orientation with ECL angle displacement. In this video, the footage exhibits relative distribution of HA labeled blue and NA labeled red and viral mobility. I was having trouble copying it into the presentation, so I'll just send you a link to my Google Doc and you can look at it for yourself. It's about 2 seconds long. This simulation demonstrates how directional movement is influenced by HA and NA polarity in a 250 nanometer length filamentous virus. The goal of the study was to characterize how HA and NA influence the mobility of filamentous virus on cell surfaces containing sialic acid, a constituent of human cell surfaces. This was accomplished by utilizing site-specific fluorescent labeling and microscopic visualization of HANA and NP. Disproportionate and polarized distribution of HANA on the viral surface resulted in binding and cleaving, which propels viruses away from their Na-abundant poles, providing viruses with the ability to both adhere to host cells and escape cell surfaces, as demonstrated in the challenge assay, which correlated increased HANA polarization with cell surface escape. The pattern of binding and cleaving sialic acid established by polarized protein distribution results in uniform HA biased directional motility. It was also determined that distribution of Na polarization was dictated by NP localization. The results discussed in previous slides were deduced utilizing quantitative and qualitative measures, which employed storm imaging, MU-NANA fluorescence, photobleaching, total internal reflectance microscopy, cell culture propagation, cell passaging, viral motility assay, and antiviral challenge assay. Limitations of this study include the inability to develop uniform models for directional dynamics and diffusion rates due to variations in viral particle shapes and sizes. Future experimentation and studies are necessary for understanding how the asymmetric distribution of viral surface proteins balance the paradoxical need to attach and detach from host cell surfaces. Here are my resources. Thanks for listening.